Good morning, ladies and gentlemen, distinguished speaker, respected moderator, and all honorable participants joining us today. Welcome to the Structural Biology for Health Research from Molecular Design to Structural Determination webinar, organized by Research Center of Biology Molecular Eijkman, Health Research Organization, National Research and Innovation Agency. We are grateful to have you all here, and we hope everyone is doing well. First of all, let us extend our gratitude to God Almighty for allowing us to gather virtually in this valuable event today. Allow me to introduce myself. I'm Andri Pramesyanti Pramono, and I will be your Master of Ceremony throughout today's webinar. Before we begin today's events, let us show our respect and love for our country by listening to the Indonesian national anthem, Indonesia Raya. We kindly ask all participants to remain in respectful position during the anthem. Let us now listen to Indonesia Raya. Mbak Rizka, please help. Okay, thank you. Now, to begin our event, please turn on your camera now and make sure your face clearly visible on screen. We would like to take a documentation for photo session. Mbak Rizka, can you help me?
Yes, Bu.
Just turn on your camera, maybe. Okay. Can take the
Okay. 1, 2, 3. Slide 2. 1, 2, 3. Slide 3. 1, 2, 3. Okay. Thank you, Bu.
Okay, thank you. To begin our event, we would like to invite Prof. Dr. DRH NLP Indi Darmayanti MSI to deliver the opening remarks. to Professor Indi. The screen is yours. Please welcome.
Assalamualaikum warahmatullahi wabarakatuh. Yang saya hormati Kepala Pusat Riset Biologi Molekuler Eijkman, Ibu Farah Novita Kautir, Ketua Kelompok Riset Biologi Struktur dan Persinyalan, Ibu Josephine, para narasumber pada hari ini, Asisten Profesor Cek Minve dari Nara Institute of Science and Technology, Dr. Yudi Nugraha, dan periset dari Kelompok Riset Biologi Struktur Pusat Riset Biologi Molekuler Eijkman, serta tentunya seluruh peserta webinar yang saya banggakan. Salam sejahtera untuk kita semuanya. Puji syukur kehadirat Allah Subhanahu wa Ta'ala, karena atas rahmatnya kita dapat berkumpul dalam keadaan sehat walafiat dalam mengikuti webinar penting ini yang mengangkat tema Structural Biology for Health Research from Molecular Design to Structural Determination. Para peserta webinar yang saya hormati, dalam dunia biomedis modern, pemahaman bagaimana mekanisme kerja biomolekul pada tingkat paling dasar merupakan kunci untuk berbagai terobosan ilmiah. Disinilah biologi struktural memainkan peran strategis karena untuk memahami, mengintervensi, bahkan merekayasa sistem biologis seperti vaksin atau obat-obatan. Kita harus terlebih dahulu mampu mengerti bentuk dan fungsi dari komponen terkecilnya melalui biologi struktur. Struktur protein tidak hanya menggambarkan bentuk, tetapi juga merupakan peta fungsional bagaimana molekul bekerja, berinteraksi, atau mengalami mutasi dalam penyakit. Oleh karena itu, investasi pada teknologi visualisasi beresolusi tinggi menjadi kebutuhan yang mendesak. Sebagai bentuk komitmen negara, Badan Riset dan Inovasi Nasional telah menghadirkan fasilitas cryo-elektron mikroskopi atau cryo-EM pertama di Indonesia, sebuah investasi besar yang memungkinkan kita memetakan biomolekul dengan resolusi hampir atomik. Selain itu, BRIN juga meluncurkan program pendanaan RIM untuk mendorong riset kolaboratif berbasis struktur molekul dan memperkuat ekosistem riset di bidang ini. Upaya ini ditujukan agar Indonesia tidak hanya menjadi konsumen teknologi, tetapi juga produsen ilmu pengetahuan yang unggul dan relevan secara global. Para hadirin yang berbahagia, Pusat Riset Biologi Molekuler Eijkman, khususnya kelompok riset biologi struktur dan persinyalan sel, telah memulai inisiasi kolaborasi dengan universitas baik di dalam dan di luar negeri, termasuk dalam determinasi struktur protein, seperti yang telah terselenggara dengan NICE berupa visiting professor dalam utilisasi cryo-EM di BRIN, serta inisiasi pembentukan asosiasi biologi struktur di Indonesia. Semoga acara webinar ini dapat menambah wawasan dan terjadi pertukaran ilmu bagi para peserta, terutama yang berkecimpung dalam riset kesehatan. Dan semoga bagi peneliti BRIN akan mendapat banyak masukan dari para senior dan peserta yang hadir. Demikian dari saya, semoga acara webinar hari ini berjalan dengan lancar. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Prof. Indi. Next, we would like to invite the head of Research Center of Biology Molecular Eijkman, Dr. Elizabeth Farah Novita Kautir, SSI, PhD. To Farah, the screen is yours. Welcome.
Ya, terima kasih Bu Andri. Selamat pagi Bapak Ibu sekalian. Assalamualaikum warahmatullahi wabarakatuh. Salam hormat dan salam sehat untuk kita semua. Yang saya hormati, Kepala Organisasi Riset Kesehatan BRIN, Profesor Dr. Dr. Niluh Putu Indi Darmayanti MSI, juga para ketua kelompok riset di Pusat Riset Biologi Molekuler Eijkman, terutama kelompok riset biologi struktur dan persinyalan sel, di mana salah satu pembicara tergabung. I would like to extend my gratitude to the speakers of today's seminar, Assistant Professor Jack Minfei, which is also called Adrian, from NARA Institute of Science and Technology, NAIST, and Dr. Yudi Nugraha from Research Group of Structural Biology and Cell Signaling at the Ekman Research Center for Molecular Biology. It is my great pleasure to welcome you and all the participants, esteemed colleagues, to the webinar, Structural Biology for Health Research from Molecular Design to Structural Determination. It is an honor for us at the Ekman Research Center for Molecular Biology to host this scientific event. The theme we will discuss today touches the core of what we believe to be the future of molecular-based health research. Seperti yang telah disampaikan juga oleh Prof. Indi sebelumnya, biologi struktural bukanlah sekedar cabang dari biologi molekuler. Bidang ilmu ini telah menjadi jembatan penting antara pengetahuan dasar dan aplikasi praktis di dunia medis dan farmasi. Ketika kita berbicara tentang penemuan vaksin baru, terapi kanker, atau obat untuk penyakit degeneratif, semuanya berakar pada pemahaman yang mendalam terhadap struktur biomolekul, khususnya protein yang merupakan unit fungsional utama dalam kehidupan. Seperti juga yang telah disampaikan, untuk mendukung pengembangan ini secara komprehensif, BRIN tidak hanya membangun infrastruktur mutakhir berupa fasilitas cryo-elektron mikroskopi, tetapi juga menciptakan instrumen pendukung berupa sumber dana, grant riset dan inovasi untuk Indonesia Maju, RIIM. Program ini dirancang untuk mempercepat riset kolaboratif dan membentuk komunitas ilmiah yang kuat dalam bidang biologi struktural. Ini adalah salah satu upaya untuk membangun critical mass di Indonesia untuk kegiatan-kegiatan riset yang berdampak nyata dan berdaya saing secara global. Saya percaya dengan semangat kolaborasi dan keterbukaan, kita bisa bersama-sama mendorong pemanfaatan teknologi ini untuk menjawab tantangan kesehatan bangsa. Semoga webinar ini memberikan wawasan baru dan membuka jalan untuk kerjasama yang lebih luas. Thank you everyone for being here and contributing to the success of this webinar. Let's make the most of this opportunity to learn, share, and grow together. Enjoy the webinar. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Bu Farah. We go to the next step. Now we begin the webinar. So I would like to introduce our moderator today. He was Dr. Syahputra Wibowo, SSE MSE. He was our researcher also in National Research and Innovation Institute in Research Center for Molecular Biology, Ekman. He also graduated from Brawijaya University in 2022, and he get a research fellowship in Università degli Studi in Siena, Italy. And also postdoctoral researcher in Gajah Mada University in 2023 to 2024. To Dr. Syahputra Wibowo, the time is yours. Welcome, please.
Hello everyone, and good morning, ladies and gentlemen, researchers, academics, and students from across Indonesia. Welcome to today's national webinar, Structural Biology for Health Research from Molecular Design to Structural Determination, organized by Structural Biology and Cell Signaling Research Group, Ekman Molecular Biology Research Center BRIN. I am Syahputra Wibowo, and I have the honor of guiding you as the moderator throughout this event. Before we begin, please ensure your microphone remain muted and your coffee stay hot, because this morning we are diving deep into science that is as precise as it is powerful. Now, let me begin with a brief prelude to our theme. In the last decade, structural biology has become a cornerstone of biomedical research. Why? Because understanding how biological molecules look and move at the atomic level is essential to understanding how diseases begin, and more importantly, how we can stop them. Throughout technologies like X-ray crystallography and cryogenic electron microscopy, or cryo-EM, scientists can now see proteins, quote unquote, enzymes and viruses in action, just like architect inspecting blueprints, but at the molecular levels. Now, these atomic level insight allow for the design of smarter drugs, targeted therapy, and more precise diagnostic. In essence, structural biology doesn't just describe life, it enables us to redesign it for a better health outcomes. And today we will hear from two leading voices in this field who are pushing the boundaries of what's possible in both global and Indonesian contexts. Let's begin with our first speaker. From biodegradable plastic to protein engineering, his work sits at the intersection of sustainability and structural biology. Currently based at the Nara Institute of Science and Technology NICE in Japan, Assistant Professor Check-Min Fei is an assistant professor specializing in X-ray crystallography and enzyme design. What makes him unique is not just his technical skill, but also his vision to engineer enzymes that help solve today's ecological and biomedical challenges. He also received international award for his contribution of protein science, and today he will walk us through the foundation and medical application of X-ray crystallography, showing us how something as rigid as crystal can be the key of innovation. Please welcome Assistant Professor Check-Min Fei.
Thank you very much for the kind introduction. Before I start my presentation, maybe let me thank all the organizer and the invitation for me to share my work today here. And also thank the Prof. Indy, the opening speech from Prof. Indy, and also Dr. Farah, as well as the moderator and Dr. Andri, and also the Dr. Putra. So let me share my screen now. Is it okay? Yeah, thank you. Is the slide all right? Yeah, okay.
Good.
Thanks. So thanks for the kind introduction. So today I'm very great to share some work in the structural biology, particularly using the technique called X-ray crystallography. Today I will just focus more on the basic concept instead of the technical part of X-ray crystallography, trying to introduce this technique to more people so that we can start working on maybe collaborative project and also try to expand the structural biology field in Indonesia. Just a little bit introduction from myself. I'm Min Fei Chet, or people normally call me Adrian as well. Like already introduced, I'm currently assistant prof in NICE, concurrently in two labs. The first one is environmental microbiology, where we discover the plastic degrading bacteria. And then I'm responsible to understand the function and also mechanism of how the enzyme degrade plastic, and also how I also working on the enzyme that produce biodegradable plastic. And another one is my concurrent lab is structural biology and protein engineering, which focus on X-ray crystallography. And both lab are in NICE, Japan. So first, let me introduce where I came from. I actually born in Malaysia, just a neighbor of Indonesia. And I got my bachelor degree in microbiology and also master degree in molecular biology from University of Science, Malaysia in Penang. And for my PhD, I have the chance to receive the Max Scholarship from the Japanese government to allow me to complete my PhD study focus in structural biology in NICE under the supervision of Professor Toshio Hakushima. And let me introduce a little bit of our beautiful campus in Nara. It's very compact. And these two are the biological science building, and we normally walk inside this building. during the weekend, also during the weekdays, and also the weekend sometime. And sometimes we also can go to a nearby sightseeing spot in Nara. For example, the Big Buddha Temple. It's called Todaiji Temple, and there is a Big Buddha statue inside this building. And so sometimes when the experiment doesn't go well, we can go to meet the Buddha inside and pray for the good result. And this is also famous for the deers, the wild deers in the Nara Park. They are being famous for polite. They will bow to you when they request the food from you. So if you have chance, please visit Nara and in Japan. So now I would like to start go to what is structural biology. Basically we can just regard this as to study the three-dimensional structures of the proteins. And now we have AlphaFold prediction. Maybe the free-form protein is not so interesting. The more significant impact we may have, maybe the protein-protein complex and also the protein-ligand complex, which will give us more information on how the protein works. And also the reason why we need the structures is because by understanding the structures, we can also understand how it works. For example, the enzyme, like I show here, this is one of my research interests. We work on the structures of the bioplastic synthase. It just published recently, and it's a first full-length structure in the world. And the right-hand side is a human protein, which is I will introduce a little bit in the end of this lecture seminar. So why protein structure is important? I would like to quote from Francis Crick, one of the big name in biology. So in his own written book, he mentioned that almost all aspects of life are engineered at the molecular level. So if we do not understand the molecules, we can only have a sketchy understanding on the life itself. So this is why the molecular biology is very important. And why it's important? Because as mentioned just now, the shape of the protein is actually important for the function. And protein is very important for a lot of biological processes. For example, they can work as enzymes and they're responsible to transport the ions and molecules. They also can work as hormones, and they also work as a signal receiver, and they also maintain the cell architecture and also cellular contraction and coordinate motion. There are so many functions. Our human and other organisms cannot live without the assistance of proteins. So before I go deeper into the X-ray crystallography, I would like just to maybe make some simple introduction on the structure itself. Protein, basically, we can separate into 4 different levels of structures. In the primary structures, we can see here, this is a polypeptide in a shorter form, and this blanket is the monomer unit of the amino acid. And how we look at this is we have the amino group on the starting point. This is why we call N-terminus or the end domain of the protein. And at the right-hand side is we call it a carboxyl group. So this is why we call it C-terminal for the following for the end of the proteins sequence. And at the end middle we have a alpha carbon, which is the key carbon that connect everything together. And the character of the amino acids is determined by this R group. There are about 20 types of different amino acids with different character, which give rise to all the different characters of proteins. And if we look at these chemical structures, and the amino acid actually coded by, we normally use a 3-letter code or 1-letter code. For example, this is a methyl group as a R group, so it's alanine, or code as ALA, or 1-letter code is A. So this is what I designed for myself. This is a peptide of Adrian, if you look at the single code. This is just for fun, okay? And then this is the R group here, and then another R group here, aspartic acid, and then arginine as R group, and this is isoleucine, alanine, and also asparagine. So this, and also the blue color means nitrogen, and the red color means oxygen. It gives rise to the negative charge and also the positive charge for the nitrogen. And after the primary structures of protein, we will go to the secondary structure. So first, I will talk about the first type of secondary structure in general. We can categorize them into alpha helix and also beta strand. Inside this alpha helix, there are also different types of alpha helix, 310 helix and different type. But in here, I will just do it briefly, introduce alpha helix as a whole. So alpha helix is a repeating patterns of local hydrogen bondings. As you can see, this is a stick representation of the alpha helix. This dashed line represent the hydrogen bonds. So you can see the R group, for example, the oxygen from the C terminal, the carboxyl group, is interacting with the amino group of another amino acid. So they form a multiple interaction between these amino acid main chain, which make them stronger as a secondary structure. So as a representative, this is just I cut out from this, so to make it easier for you to understand. And this is also a cartoon representation so that you can see is the NH, which give rise to the partial positive charge, and O is a partial negative, so give rise to the interaction between these. And we call these hydrogen bonds. And another type of secondary structure is called beta sheet or strand. Is actually a repeating patterns between the distant part of the polypeptide chain. So we never see the beta strand as single strand. The reason why is because the beta strand need to interact side by side. So this is how we can see, we normally call the strand like anti-parallel or parallel strand. So the direction of the strand, like one up, one down, or at the same direction. So you can see the interaction also same. They have the hydrogen bonds between the O and the N, but this is at a different conformation compared to the alpha helix. So now we already know the general secondary structures. Next, we go to the tertiary structures. Sorry, before that, let's go to a fun fact of the history. When did they discover this conformation? It's back in 1951 by Linus Pauling, the giant in biological science from Caltech. And this is the first paper on the helix structures based on their calculation, and it's proven to be correct after we have the structural biology and looking at the structure. And the right side is the beta strand configuration. So if you're interested, you can look at the old paper and see how they describe the alpha helix and beta strand. For the tertiary structure, to make it simple, it's just a combination of everything: alpha helix, beta sheet or strand, beta strands together. And then how they connect to each other is by loop. Loop is some chain that do not have a specific structure. And the importance of the tertiary structure is that they bring the important amino acid together. Because in the primary sequence, it's just a one line of sequence. We cannot see the important part, but in the tertiary structures, they bring the important part come together. For example, enzyme activity or binding pocket, or the important interaction site of the receptor and also the activator. So this is the pocket formed by after we form the tertiary structure. This is the information that we cannot get when we have only the primary sequence. And this is formed by through a lot of chemical interaction, include the hydrophobic interactions, hydrogen bonding, and electrostatic forces. So hydrophobic is literally they do not like water, the amino acid that do not like water. So to avoid the contact with the water, they tend to fall into inner core of the proteins. So it makes it a quite strong interaction for proteins. Then the next highest level, we call it quaternary structures, which means more than one single polypeptide chain. We can regard it as maybe dimer. Homodimer is also one of the example of quaternary structure. Or we can have the example of hemoglobin, which is consists of 4 different polypeptide chain with a 4 hem group. this is the 4 levels of the protein structure. And in summary, or just a recap before I go deeper into the protein structures. So we have already introduced primary, secondary, and tertiary and quaternary structures. And proteins generally involved in a lot of various biological processes, and the shape and the protein and the structure of protein are important for understanding the structures. And structural biology is important to review the function by determining the 3D structures, which are the information that we cannot obtain from the primary structure itself. So how do we get to the determination of the protein structures? Currently, the 3 major methods scientists use are MMR, nuclear magnetic resonance spectroscopy, X-ray crystallography, as well as cryo-EM, cryo-electron microscopy. So I just introduce some limits and also the merit of every methods. MMR is very good to capture the dynamic movement. However, is limited to small proteins, for example, smaller than 25 kilodalton, which is too small for most of the human proteins and also the larger enzymes in real life. And for X-ray crystallography, the merit is it's a very established method, and it can achieve atomic resolution. And there's no limit on the molecular weight. However, you need to have the crystal, which is one of the most difficult part. And relatively new technology is called cryo-electron microscopy, which just received a Nobel Prize recently. The merit of this cryo-EM is that it is relatively easier. You do not need a crystal. However, the technology might need larger protein because it's microscopy. Now the molecular weight breakthrough already go down to much lower. I think Dr. Yudi will explain later. And another limitation is require the powerful graphic processing system because we need to identify the small dot in the micrograph. However, I think BRIN already equipped with all these advanced system, so it's very good platform for the scientists in BRIN to utilize a cryo-EM technology, which will explain by Dr. Yudi later. And now, after we determine the structures of protein, normally we will upload or deposit the structures into a database called Protein Data Bank. And we can, this is all the database for the experimental determined structures of proteins or protein or DNA, protein-DNA complex. So once you go to this website, there's a search bar here. You can type the PDB ID or the name of the protein that you want to search. And in the PDB, all the structures have a PDB ID code. For example, 63W1 normally come from a 4 code. However, recently, because the code is almost full, I think they will expand the numbering or the ID of this PDB. And just an overview of the structures deposited in PDB by different methods. Up to, I downloaded this statistic yesterday, 2 days ago. You can see that initially it's all by X-ray crystallography since the old time. And after 2020, you can see the cryo-EM continue to increase every year. And this year we just passed 6 months, so it's still a lot to go. I'm add on for the year of 2025. So we can see that the trend of the structures determined by cryo-EM is increasing now every year. So today I will focus on the X-ray crystallography, and Dr. Yudi will explain more on the cryo-EM part. So for the X-ray crystallography, I will explain on the methodology, but I will try to make it more general and by introduce some concept instead of technical details. So in the overview, normally we can separate into a few steps. For example, the molecular cloning and design of the construct. Then after that, we will go to the expression and purification of the protein, one of the key steps to obtain the super pure proteins. And then we go to another bottleneck difficulties, which is trying to crystallize the proteins. After that, we will send the crystal to collect the X-ray diffraction data. Then only we can collect the data and try to determine the crystal structure using the data diffraction information. Once we have the enzyme or the protein structures, we can start to analyze the protein structures and try to understand and interpret the importance of these structures. Because if we do not give the meaning of the structures is just a cartoon without anything. So this is the important part for structural biology to understand and what is the importance of this structure. So this is just a simple introduction of the gene construct and protein design. Before we have the structure, this is in the old time. I didn't update after the AlphaFold appear. So in the when we start to do this, we normally obtain the sequence, for example, the protein sequence. Then we try to compare the sequence with the homologs that we can find in NCBI database. And we also can predict the secondary structure prediction. Why we do that is because to obtain the good crystal, sometimes we need a stable protein. The flexible region we need to remove. So by looking at the secondary structures, we can truncate or remove the non-conserved region, or we can remove the loop region, which is a non-structural part. And we also can introduce a mutant to increase the protein stabilities. After we finish the cloning, and then we can start to go to the next step, which is to express the proteins and purify the protein. So how we normally do this is that we will use the organism to produce the target proteins. Mostly, if it's an easy protein, then normally we can just use microbes like E. coli. However, if it's sometimes it's a human protein that require the post modification or is important, it's very difficult to use microbes. Then maybe we need to use a mammalian expression system. So, which is, I think, Dr. UD is also trying to set up the system for mammalian protein expression in BRIN. So I just introduce a flow that we do it here. So we can see this is the large-scale expression of the E. coli. So about 12 liters per batch of the culture. And after that, we will harvest the cell, pellet, and then lyse the cell, collect the protein because they produce a protein. Then we will use the ultracentrifuge to separate the insoluble part and the soluble part. So this is just a clearer image for you to see. This is come from maybe 6-liter culture or 12-liter culture. You can see the milky color of the bacterial cell suspension. Then after the sonication, we will see the clearer solution, and then we separate the insoluble part and the soluble part by going through the ultracentrifugation. The purpose of this step is to separate the like wrongly folded protein or non-essential the cell debris from the E. coli. So after separate, we will use the supernatant to load into the purification system to collect our target proteins. So this is just an overview of the multi-step chromatography that we can use to purify the protein. The starting point, we can see there are so many bands in the total protein lysate come from the ultracentrifuge supernatant. And after the multi-step purification, we can see, and we hope to get a single band protein purity higher than 90%. So the common step that we can use is first affinity chromatography, which is by using the recombinant technique. We can purify using the resin. And then we can further purify using ion exchange chromatography by separate the protein based on the surface charges of the protein, maybe negative charge or positive charge. Then finally, we normally polish the purity of the protein by size exclusion chromatography. In this step, the size exclusion chromatography is normally separate the protein by sizes. So every step have different purpose. So it depends on how we're going to use it and what is the character of your target proteins. And we tend to reduce the step of the purification because in each step you lost maybe 30 to 50% of the protein amount. So this is the reason why we normally start with the large amount of starting protein, because you will lose the quantity to achieve the quality and purity of the proteins. And now I will go to the next step, which is crystallization. I think this might be new to a lot of audience. So I will go a little bit slower here. And I will explain why the purity of the protein is important for crystallization. The most common technique in protein crystallization is called vapor diffusion technique. So you can see now I'm using these two examples. So this is the precipitant. The protein drop normally is represent using this green color drop. So this is called hanging drop. As you can see, the drop is hanging on the cover slip. Another technique is called sitting drop, which is the drop is sitting on a well. And in both condition is a closed condition. And then there's a precipitant represented by the blue color. They absorb water from the environment. So since this is a closed environment, you can imagine that the water molecule will leave the protein drop and go to the precipitant condition solution. So during the process, different precipitant will have different speed of absorption. So by doing that slowly, the protein will be more and more concentrated. So when it's more and more concentrated, the protein will tends to align to each other, and then hopefully they form a uniform order and then become a crystal. And this is a basic step that we use. We use a Mosquito automated liquid handler. It's a robot machine that helps us to set up the screening kit. If we set up by hand, maybe we take 1 day to set up 1 kit. But using this, we can reduce the time to 30 minutes for 1 kit. So it's much more efficient and increase the chance of getting a good crystal. I think this is also preparing in BRIN to enhance the chances of crystallization trial. After that, we will set the protein crystal screening in 4 degrees and 20 degrees Celsius in the incubator. Then we observe the protein growth, protein crystal growth by observing using the microscope after every week or so. Now I will go into slightly more details on how it works, the hanging drop. So in this drop, the green color drop is come from a protein and crystallization solution. And then in this blue color drop is the crystallization solution, which is the precipitant that I mentioned just now. And H2O will be absorbed into the precipitant. After a few days of equilibration and evaporation, the drop becomes smaller. So you can imagine the concentration become higher, and we will form the crystal inside this small drop. This is just for your visualization. We look from the top of the screening plate. The drop is on the top of the slip, and this is the crystallization solution at the bottom of the well. And the condition is sealed so that it will just absorb the water from the crystallization drop. And this is how we look at it, the chart. When the protein concentration is low, it assists in the nothing will be appear. It's just solution form. When the concentration increase, it slowly go to the nucleation zone. Then they form the crystal crystal form by aligning the molecule at a uniform order. Then they continue to grow to the crystal. However, if the concentration is too high, they will precipitate. And if the concentration is too low, they remain as solution. So this is where we need to play around with the protein concentration and also the precipitant concentration to find the right condition to get the good crystal. And how the crystal form here, I would just like to introduce a little bit concept on how the crystal grow. As I mentioned just now, the initial crystallization drop is just a solution. So this water protein molecule is just freely moving around in the solution. When they become more and more concentrated, they have higher chance to meet each other. And we hope that it will align properly like something like this. So when it align properly like this, it's actually forming the crystal. So this is all the crystal I obtained during my PhD study. You can see this one is a good crystal because it's single, but this one is not, it's cluster. So we need to optimize this crystal to become single so that we can get a beautiful diffraction data. And this is just to show you that inside one crystal, actually there are not only one protein. There are millions of proteins inside aligned together like this. So this is one of the example of the protein that I solved. This is probably the smallest symmetry unit. So you can see this is aligned together at a uniform order. So in one protein crystal. And also in the protein crystal, there are about 50% of solvent. It's not dry, it's actually contain water. So this is some of the misconception. People will think that the crystal structure is just dry, but this is a macromolecule crystal, so it's different from the inorganic crystal. It contains about 50% of water inside the crystal, and which make it very difficult to do. So why protein purity is important? As I mentioned, we need the protein to form a proper align uniform orders. So if they have a flexible region, like at the end terminal or some part of the protein, it might actually disrupt or disturb the alignment of the proteins. So this is why we have to design the protein to make them more compact and remove the flexible region. And why purity matters? Because if there are all impurities in the solution drop, you can imagine some other protein or compound might disturb the alignment and formation of the crystal. So once we have the good crystal, we can go to the final step, which is another bottleneck in this method, to collect the X-ray diffraction from the crystal. So here in NICE, because our university is actually quite close to one of the synchrotron radiation facility, super photon ring 8 gigawatt is called Spring 8 in short. It's an electromagnetic wave entering from here, the emission region, and then it will accelerated by the bending of the magnetic field and get close to the light of speed. And then when we do the experiment is we will share some of the beam into this called beam line, and we perform the experiment inside this hub. So we will set the crystal inside this beam line hub, and we control the measurement outside of the hub. And this is just some of the picture of the whole Spring 8, which me and UD went when he studied a PhD in NICE. And how we harvest a crystal, this is also requires some technique. We will use a loop to collect the crystal. For example, we will scoop the crystal by using this loop under the microscope. So it's also quite challenging and for beginner. So you need some time to practice to learn how to collect the crystal. So after we collect the crystal loop, you can see inside this loop, this is a crystal. And here is where we bombard the crystal by the X-ray beam. And this is the machine inside the X-ray hub that I mentioned just now, in the Spring 8. So the X-ray beam will come from here and then hit the crystal, and it will scatter the diffraction spot and place all the diffraction on the detection. And we will not only collect the single frame, so we will rotate the crystal for maybe 360 degrees, so that we can see the whole thing from the X-ray diffraction data. So this is some of the, if you can look at the, I'm not sure whether it is work for your side. I worry about the internet. So if you can look at the rotation, this is all the spot representing the electron density from a crystal. So when you rotate, you can see the spot is moving in a uniform way. So we will use this information from the diffraction spot and convert back to the 3D structure. And this is inside the spring 8. The ring, we can actually use the internal bicycle to cycle for maybe one round about 10 minutes. So this is all the beam, different scientists working on the spring 8. Like not only biology, there are a lot of material science scientists. For example, Toyota company also own one beam line to study their own materials. Okay, so now I would like just to recap a little bit on the flow of the data collections. So from the crystal structure, from the protein crystal we bombard to X-ray, and then we collect the diffraction pattern. Then we will perform a Fourier transform and phasing to generate the initial electron density map. Then we will fit the protein models into the map following based on the electron density map, which will make the structure model. And this is the final 3D structure determined by doing this process. And this is our assistant professor teaching the student, which I collect from the back. I think they didn't know until today. And just an example of the in the history, the first protein structure from X-ray crystallography is actually reported in Nature journal back in 1958. So this is maybe the picture doesn't look very nice in the morning, but this is actually the structures determined from sperm whale myoglobin. And the resolution was subsequently improved by another big name in crystallography, Max Perus, which is also published in Nature back in 1960. The reason why they use a sperm whale myoglobin because back then the recombinant protein purification technique, there is no such technique, so they cannot purify the protein in larger scale. So what they did is, because sperm whale is big, and just collect the protein from the blood of the sperm whale. So this is the abundant protein they can get. So they use this as a target to study and initiate the X-ray crystallography technique. And finally, I will go to the structural analysis. So in the structural analysis of protein, once we got the structures, we can actually try to understand like what amino acids are on the surface. For example, the blue color is positive charge, and then the red color is negative charge, and the white color is hydrophobic interaction. So when you look at these two, it's actually same protein, and we know that this peptide bind to this region. So this is white color. We can know that this is a hydrophobic interaction between this compound. So what we can do is we can actually look at how they interact. We can improve by doing mutation to make the binding stronger. We may be used as an inhibitor. We will block the natural binding between, for example, virus, how virus attack the virus response protein in the human. And then we just design some protein that can block the binding of the virus as an inhibitor, which will become a drug. And also we can understand the mechanism by identify the catalytic residues of the enzyme. So this is the geometry is important, and we can perform the engineering to enhance the enzyme performance. And the oligomer status, we also can know, for example, how the protein-protein interact to each other and the protein ligand interact to each other. And this is what I say, once we know how they bind, we can actually modify the ligand to make them bind stronger. to improve the affinity of the drug. For example, we screened the natural product in Indonesia, and then we found that this drug actually has a function to inhibit certain protein interaction. We can actually look at how they bind and then improve the drug affinity by redesign the drug once we know the structure, which will increase the high affinity of the drug so that we don't need to subscribe like 10 milligrams of the drug. We can reduce the drug concentration, which will be better and more specific to the patient. So there's all for the introduction of the X-ray crystallography. Now I would like to introduce the history of another drug scandal, one of the biggest drug scandal in the history, and also one of the project our lab is working on. It's not my main project, but I will present what our previous assistant prof working on. It's called thalidomide drug. This is a drug that caused over 10,000 of birth defects back in 1960s. So this thalidomide is marketed in 1957 and is withdrawn on 1962. It's a drug that market for the morning sickness. However, it unexpectedly causing the birth defects, especially the limbless baby. And however, after the withdrawal of the market from the market in 1960, the scientists found that there are positive use of thalidomide in treating leprosy and also multiple myeloma, which is approved by the FDA in 2006. However, nobody knows why and how thalidomide will create such a big problem. So this is how the structural biology came into play. First, the target of thalidomide was finally discovered by a group of scientists from Japan, from Handa Sensei Group, which is published in 2010 in Science. So what they found that is when there is no thalidomide, the chicken can grow the limb normally. But when there is a thalidomide, they found that the limb was shorter. There is no limb compared to the without thalidomide treatment. And in the paper, they describe how they discovered the CRBN, which is the target for the thalidomide. So what is cereblon CRBN? CRBN is a E3 ubiquitin ligase, and it's a CRBN autologs are highly conserved from plants to humans, which means this protein is very important for a lot of organisms. And CRBN is a putative substrate receptor. So this blue color CRBN, this is the protein that recognizes the substrate to be digested or degraded. And so far, there are 4 putative targets of CRBN have been identified, and these 4, which is a natural product or natural substrate of CRBN. After they recognize the substrate, this is the whole complex of CRL4 complex, and then they will ubiquitinize the substrate. Once they are ubiquitinized, they will be recognized by the proteasome, and the substrate will be degraded. So this is how the body control the protein level by using this system. However, nobody knows why. So after that, scientists have tried to identify and understand the mechanism by determining the structures. This is a very hot topic back then. In 2014, 2 independent groups have published a paper in Nature and Nature Structural and Molecular Biology. So this is a collaboration work between NICE, and I forgot which university, but Hakushima Sensei, which is our PI, is the collaborator of this project. And also the main person who did this work is Tomoyuki Mori, our previous assistant professor. So in the structure, they determine the complex of DDB1, so this one, and also the CRBN here, and to recognize how they bind to the thalidomide drug. So this is just zooming into where the thalidomide binding part. So we can see that This is thalidomide, the yellow color. So thalidomide consists of all these three different drugs. Show the similarity in the glutalimide ring here, the red color coded, and the difference part is at the thalidomide moiety. So this part is different, and you can see this is the thalidomide complex, and then lenalidomide complex, and also pomalidomide complex. What made them interesting is that we realized, in the following study, we realized that after thalidomide bind to this CRBN, it actually shifts the target to non-natural substrate, which means they digest what should not be digest. And this is why the baby limbs was not grow, because once you ingested the thalidomide, it actually shift the protein digestion process to another substrate that is important for the growth of the limbs. And then when they use a lenalidomide and pomalidomide as the substrate, they realize that they actually change the substrate, which means for example, natural substrate is MEIS2, but once they incorporate the lenalidomide, they actually digest Icaros, which is not the original substrate. And they call this immunomodulatory drugs, or IMIDs, which induce the ubiquitination and also proteasome degradation of the key factors Icaros 1 and 3, which is IKZF1 and IKZF3, and which is also proven by the structural determination in the following in 2018. So currently, this discovery has a lead to the drug design based because, for example, once with using this different drug, the substrate can be shifted, and they call this method PROTAX, which is a proteolysis targeting chimera, targeting protein degradation. To treat some protein, there is a difficult to use inhibitor. So by using this method, it can actually naturally digest or degrade the protein in the body. So I think this is just some brief introduction. If you're interested to know more, please ask me later. And before I end my seminar today, my sharing today, I would like just to introduce a little bit on our lab. It's a lab of structural biology and protein engineering. And our PI is Toshio Hakushima Sensei, who is in the middle. And if you are interested to know more our lab, please Google NICE Hakushima Laboratory. And I was trained in this lab, focusing on structural biology. And when I graduate, I think UD joined our lab as a PhD student as well. And this is an acknowledgement to this project, mainly done by the Professor Hakushima lab. And our mentor who teaches all the technique is Assistant Prof. Kim Sang-yong, who is this very kind man. He teaches all the technique and is a very good teacher for us. I mean, me and Dr. Yudi. And also, currently, I'm affiliated with the environmental microbiology in NICE with Professor Shosuke Yoshida, who is a discoverer of the plastic degrading bacteria. And this is our lab. Sometimes we have some party, so this is the party picture, and this is UD when he is still young, and we can go to climb mountains during our weekend. And this is just for some advertisement. If you're interested to know more about bioplastic synthase, please feel free to have a look. Recently, we published the first full-length structures of bioplastic synthase, which is published in Angewate Chemistry Journal. So if you have an interest, please. And with that, I would like to thank everyone for listening. I'm ready for any discussion or question. Thank you very much.
Thank you very much. Dr. Check or Dr. Adrian for such a
Yes.
clear and compelling talk. So before we going to the Q&A session, I would like to remind again for the webinar participant to get the webinar certificate, you should fill out the online attendance form on the chat section. So we'll now open the floor for the question. If you have something to ask, please use the Q&A feature or raise your hand virtually, and we will give the time. We welcome both technical and practical questions as well. Is there any question? Okay, maybe to start the question, I will ask Dr. Check, what do you think about the phasing problem? Because we all know that the X-ray crystallography has a certain problem with the loss of structural determination, like phasing problem. If we have the really high angstrom, like under than 1.2 angstrom, we can use the direct method, right? But what if we had not so clear for the protein? So
For that problem, I think it's not too much on the resolution. I think nowadays, I think you still can try to get the molecular replacement by using for example, we can just extract the core region or the stable domain as a search model to try to solve the structures. And another way is that if you do not have the similar structures, then maybe we can try to use AlphaFold or other prediction tool to help us. They actually give us some good result during the molecular replacement part, which is very helpful to solve the phase problem. But I don't think it's a very major problem now, unless your protein is very, very unique. Then maybe it's a problem that we might need to go back to the old method by using the single anomalous or multiple anomalous method to try to solve it by using experimental determined structures with the heavy metal. Yeah.
okay.
Thank you for the question.
Thank you. Okay, Mr. Yudi.
Thank you very much, Dr. Adrian, for the great talk. My name is Yudo from Kyoto University. And my field is virology, and I'm also very much interested in structural modeling, especially for quaternary structure or binding between viral proteins, like like envelopes with the host receptors.
Yes.
And while the residue-residue interaction is, of course, very insightful for us to know the binding pockets and stuff, but we also, especially recently, we know the roles of post modifications. I think you also mentioned, like glycosylation or, for example, in HIV envelope, the glycosylation and conformational changes, and also lipids, like in hepatitis B. There's a myristoyl group on the N-terminus, which affects the membrane binding, and it now found to be very important. So I just wonder, because we use mammalian cells as well for the expression factor and to get this binding to work. But in the process, which is I'm not familiar with, from crystallization and or for cryo-EM, whether these post-translational modifications, how well can we infer from the finished model on these modifications. And the second question, if you don't mind, you mentioned about AlphaFold, but I also noticed for the again the quaternary modeling using AlphaFold multimer. I heard that it's very computationally heavy, but I just wonder whether how good is it based on your experience and knowledge to actually model this binding protein binding. Thank you so much.
Okay, thank you very much for your question. The first question is on the post modification. So for the post modification, I think that it depends on what type of post modification you want, and also what type, because sometimes after the post modification, for example, glycosylation or phosphorylation, the structure may change. So in that case, maybe it will be better for us to use the in vitro method. For example, we prepare a protein using the phosphomimic mutant and also non-phosphomimic mutant, which is we can see the structure before the phosphorylation happened and also after the phosphorylation happened. Then we will have two different structures, which will help us to understand the whole mechanism. And another one, for example, the glycosylation and also maybe acetylation, we can produce the protein using the mammalian cell and then let them naturally post-modify. And after that, we can view it maybe under the cryo-EM or we crystallize it. Then we can see, actually, when the resolution is high enough, we can see where is the exact post-modification compound, like the acetyl group or the glycosylation site. So it actually depends on what is your target proteins and also what type of post-modification. And you have to be careful that in some case, if you use a mammalian cell produced protein, it's actually a mixture of post-modified and non-post-modified species inside. And this is not very good for crystallization because you have two different proteins and probably two different conformation of protein, which will disturb the crystallization process. And in this case, maybe UD will mention about the cryo-EM technique, which has a slightly dynamic conformation, can maybe categorized during the image picking. So we call this digital purification. So you have two different types, open form and closed form in the cryo-EM micrograph. And we use the GPU in the software to pick up different conformation and separate into two groups. Then in that case, maybe it's much easier. But it depends on how different are they. If the difference is too small, I think it's still difficult to be separated by cryo-EM. So this is for the first question. And then for the AlphaFold modeling, I think it depends on how well the template or the similar protein are in the PDB, the protein data bank. Because AlphaFold is actually trained by AI, which is using the proteins inside the protein data banks as a training sample. So if there is not much sample in the PDB, they cannot train well, right? So for now, I think the modeling is still not, for the multimer, for the oligomerization is still not very high accuracy based on what I know. But for the modeling based on the freeform, which is already well-established protein in the protein data bank, then the accuracy can be quite high. So anyway, we just have to be careful after you got the structures. We just have to judge with caution. But it's very good tool for us to make hypotheses, and also maybe a good way to let us expect what we might get by the end of this project. I think it's very useful for us, and also useful for us to solve the structure, especially the dealing with the phase problem, like Putra-san mentioned just now. Yeah, and also maybe when writing the grant, we can have some estimation, like the expected result. I think this is one of the good tools, but we still have to use it cautiously. I think that's my answer to you.
Thank you very much for the answer. So in the chat room, we have several questions here from Mr. Rois. He said that many protein structure appear to exhibit symmetry. Does this symmetry arise naturally, or is it primarily a result of the crystallization process used during structure determination?
Oh, I see. So this is a natural result of the crystallization process, and it depends on the crystallization condition. So one protein may have a different symmetry or space group, we say. So it actually depends on what chemical you use for crystallizing them. like I explained just now, the crystallization solution. So it's actually depends on what condition. But if you use the same condition to crystallize a protein, then it should be the same space group. Okay.
Okay, then Mr. Agus also asks about the how which thalidomide cannot cause effect of teratogenesis that we said before. Maybe he
Oh, I see. So as I mentioned just now, maybe so if you look at this thalidomide, you can actually see the differences happen between the thalidomide moiety between these three study drugs. So how we can do this is actually we can play around with this part and see if we can shift the teratogenesis process impact of this thalidomide domain. So I hope this is the answer for you.
okay, then maybe this is a little bit technical question from Miss Aura.
Okay.
She asks about are cryo-EM micro-LED typically integrated with X-ray crystallography as part of a combined approach, or are they considered entirely separate methodologies, especially for analyzing small molecules?
I see. I prefer to combine because they have a different strength. I mean, cryo-EM has its own strength, especially good for virus like dengue virus, malaria virus, and also the big protein like maybe membrane protein, which is a very difficult protein to be purified because they are positioned at the membrane region. So it's quite expensive to produce them in large scale, and it's also not easy to crystallize them because of the presence of detergent. So during that case, a lot of people use cryo-EM, especially for the membrane protein scientists. They shift from X-ray crystallography to utilizing the cryo-EM method, which is much cheaper and also much more efficient for their field. And for the X-ray crystallography, I think it still remains because, for example, using the enzymology, it still requires the high resolution. For example, we need to see how the ligand bind. So if the resolution is high enough, then we can exactly see how the ligand go into the binding pocket, which is very good for the design or protein engineering. So, but however, if you mention about the small molecule, maybe the cryo-EM might be quite difficult. You might see something, but to until the determination step, I mean, the process might be quite difficult. For example, I will show some picture here. So this is my protein that I use crystallization. It's easy to crystallize for not easy, but I already found the optimized condition that can crystallize my protein of interest. And the right side is a cryo-EM image that I obtained with Dr. Yudi at the BRIN Cibinong cryo-EM facility. You can see we can actually see something, but due to the size is smaller, so we might need more modification or the optimization in the computational part. So I think you can try both. If it's difficult to crystallize, then you try cryo-EM. But if your protein is big enough, maybe you just try cryo-EM directly. Yeah, I think there's the answer.
Okay, thank you, Dr. Check.
Thank you.
There is, if you have time more for these two questions, the first one from Miss Ella. She asks about, do you have any tips or trick to capture the structure of enzyme in the intermediate form, which is while binding with the substrate? For example, to further engineer the enzyme to increase product specificity.
Sure. Yeah. Hi, Ella. I think I met you last time in March. Thank you for the question. And I think what we normally do is you can have several methods. The one is if your ligand is small enough and and is easily accessible to the surface of the protein, then maybe one method you can use is called soaking. Once you have the free-form crystal, you just insert the ligand and then let the ligand go into the pocket of the crystal that you already formed. Another way is we normally cold crystallize, which is one of my preferred way. However, you need to have a higher concentration of the substrate. For example, you use 0.3 millimolar of protein. Then normally we try with 10 times or 20 times more. For example, 6 millimolar or 3 millimolar of the ligand. So you try to force the ligand to go into the correct pocket. Then you try to screen for the crystallization condition. And this is how I do it. Why? Because when they are in the intermediate form, I think sometimes they can actually stabilize the protein, which is good for the crystallization process. So when you cannot get the free form or upper form enzyme structure, actually you can just try the complex form. Sometimes it will be much easier. Yeah, then it's good information for us to engineer the enzyme.
Okay. Thank you, Dr. Check. Maybe the last question.
Okay.
So there is Dr. Ita. She has 64 TDA tetramer protein to be analyzed by cryo-EM. Maybe this is for Mr. Yudi because this is EM, but then maybe she just want to ask you, what are the purification and condition requirement for crystallography before cryo-EM? Is
Okay.
there any alternative to combining with another protein to make it large enough to be analyzed by cryo-EM? And what are your suggestions for the type of protein? Because after the combination, is the nature of the original protein not changed by that combination?
All right.
Okay.
Okay, thank you for your question, Dr. Ita Margareta. And okay, 64 kilodalton is a monomer size or tetramer size? If it's tetramer size, then maybe it's very small. But if it's monomer size, then if your protein is tetramer, then it may be 200 kilodalton. They will be very good for cryo-EM. But in the case, okay, it's a tetramer, so it's quite small for cryo-EM. I think the purification and condition required for crystallography and the cryo-EM is the same. We want to get the pure protein as much as possible, and the protein purity will affect the quality of the structure that we obtain. So either way, we need a pure protein and also I would say homogeneous, which is in the single form. For example, open form or open form, closed form or closed form. It will be much easier to get higher resolution data. And you have a good question on if you increase the size of the protein. Yeah, this is also one thing that we might work on together with BRIN and especially Dr. Yudi and the team. We are trying to think on how to increase the size by designing some tech. And it's actually somebody already did, but the problem is we need a stable form. Like what you say, one thing may become concern is that the nature of the protein might not change or might change. We're not sure. We have to try and see. But I suppose they will not change that much because we often use the fusion tech to purify the protein. If the tech affect too much, then maybe nobody will use that technique anymore. But it's a very common technique nowadays for purify the protein. There's one thing, but it actually depends on the nature of your protein as well. Because your protein form tetramer, if we increase some tech inside, maybe it will affect the tetramer formation. Or, for example, we put the fusion tech at the N-terminal, and the N-terminal region might require for the tetramer formation, then this is the thing that we need to be careful. So this is the good thing where you can try to play around with prediction. Maybe you just include some tag and then try to run the maybe alpha multimer, like the Yodi Opini mentioned just now, and then see how did they still form a stable tetramer like without the tag. So in that case, maybe you can reduce the chance of failure and enhance the chance of success. However, now I think there are still limitations, especially after you connect the tag. The linker between the tag and your protein may affect, because in cryo-EM, when we try to capture the image, it should exist as a SAM, as a whole. But you can imagine if the linker is moving around, one image is here, one image is there, one image here. Maybe the fusion tag, you can get high resolution, but the local resolution for your target protein maybe is messing around because it's keep moving. You can imagine when you take the picture of the kid running around and you use slow shutter speed, it blur everything because they keep moving around. So you will get a low resolution for the local part of your target protein if the linker between the fusion tag and your protein is not properly fixed. There's a to answer your question is possible, but I think it's still not mature yet, the technology. Thank you.
Thank you, Dr. Check. Maybe that's the end of our Q&A session for
Thank you.
the first session. Now we are moving to the second session. We are honored to hear from someone leading the frontliners of structural biology in Indonesia. Dr. Yudi Nugraha is a structural biologist and biomedical researcher at PRBM BRIN. He also the head of scientific committee for Indonesia first cryo-EM national facility. His experience spans postdoctoral research in Spain and Italy, biomedical innovation, and numerous publications across top journals. But more than credential, what stands out is his commitment to building Indonesia molecular research infrastructure, making high-resolution cryo-EM accessible not only for the elite labs, but for the national scientific community. Today, he will guide us through the world of cryo-electron microscopy, a tool that allows us to see the unseeable, especially complex protein, viruses, and membrane-bound structure that are often elusive to the traditional methods. Please welcome Dr. Yudi Nugraha.
Thank you, Pak Putra, for introducing me. Firstly, I would like to thank Prof. Indi and also Bu Farah to initiate this webinar to attract people working on structural biology, starting from research of organization of health here, and also I'm happy to meet you again, my senpai, now becoming sensei in nice, Dr. Check Min Fei. Yeah. People usually confused with calling you Adrian and Check Min Fei. So I'm really happy because I'm learning also to refresh me about the structural biology, working with the X-ray crystallography, that the thing that I already far from to 2020 2021. So I would like to share my screen here. Okay, I hope we can see this screen, right? So I'm belong to structural biology and cell signaling research group led by Dr. Josephine, and also under Research Center for Molecular Eggman, now under BRIN, led by Dr. Farah. And also we are working together to establish the cryo-EM lab since 2022 and 2023 we established on December. Nowadays, so many people working with this in our lab, even like full until August for working with the 2D imaging, but not for protein purposes. So we remain still lack of the protein sample with this. So that's why the initiation is very good here to start with this webinar. So I think this I have several sections here, maybe like 5 sections, but the first until 3 maybe already very well explained, crystal clear by Dr. Adrian, related why structural biology. I show here only to say that it is very important, and also the end of how molecular biology work. Many people of us in Eggman, especially working with the molecular biology, very much about engineering, and also many working with the to understand mechanism and also the vaccine mRNA and so many things another that we cannot say like epidemiology also. But mostly it is like we will touch another DN of the molecular biology is a structural biology, but it's also the starting point for engineering of molecular biology. Mostly that we know that the largest number of Nobel laureate is coming from our discipline. It's under structural biology. So I think also Dr. Adrian already told this about dogma centra in biology. The short thing that we can say that our body, of course, like code by DNA, but the machinery of it is translated to be the protein that consists from the amino acid as the as the block chain of the protein itself. So the defect of, for example, mutation in the DNA actually defected to the molecular machinery in the cell, of course, is by the protein. Protein is itself is one of the 4 macromolecule that have very conserved structure compared to another. So this structure is related always as a function. So the defect, for example, of DNA, it will be also defect of one, for example, amino acid. This defect will also have anomaly in the structure of the protein, which make a very serious problem in our health. So I think the easiest way to know how the structural biology work is the watch. Inside there is so many machinery. We can like an analogy here as a protein. So the defect of one protein make a cascade of the malfunction of our body. So we also understand that the protein is very selective to their ligand, including their ligand as a protein, also drug. So understanding the structure to from the will be like lead us to understand the function. It is like just remaining in the introduction. So, but in the future, I think the manipulation and engineering also very much based on the structure. So it is we call it structure-based drug design for drug. So we have already enjoy the explanation from Dr. Adrian related to how determine the structure by one of the most famous one, the X-ray crystallography thing. And also now I continue to cryo-EM. But first, Dr. Adrian also already mentioned how we work starting from the protein target identification. As we already now we can like harvest from the gene bank and also to understand the construct the very stable, and we put the gene cloning and do the expression and purification. It is very hard work. Some people confuse with the structural biology in Indonesia working with the bioinformatic and also in silico only, but also we both. We use half of this in front of computer, but most of them, about like 80%, working with the wet lab in laboratory. So after structural determination, structural biology also working with the development. After understanding the structure of protein, for example, we can like understand how the mechanism, and then we can do our purposes, for example, to inhibit or also to like design the drug. Later in another section, I will show you the case that I have the experience working with of SARS-CoV-2. At that time, very famous, the pandemic. That's why I went to Madrid at that time. So why cryo-EM? Because we already understand that the number of the PDB structure published in deposit in the protein database is very huge from the X-ray until now. But the trend we can see after we can see the trend of the EM electron microscopy is in the exponential pace. So it is like people now mostly shifting from X-ray crystallography to EM. The sample reduce from the very much. For example, we need to express. Dr. Adrian said in one batch like 12 liter. because we need, for example, resulting from 12 liter is like a 12 milligram, and for example, and then for crystallization we need, for example, at least 100 microliter to optimize this huge money and huge cost for making the sample. But in cryo-EM, we only need like a 4 microliter sample is more than enough in our grid. For optimization, maybe like several five or four starting in the vitrification is also enough. But the most reason people going to cryo-EM is some protein cannot be crystallized in like it is not always protein can be crystallized. Even now in our BRIN itself, we're starting build the facility for crystallization robot in our and the management also still under cryo-EM. But please be like understand that the not all protein can be crystallized, but it is like why people going to the cryo-EM. And also, as Dr. Adrian mentioned before, we do the optimization of construct. Also, for example, I got my PhD working on PPER. We make several construct avoiding like a coil part of the protein. The construct we delete many, and we have at least like a 5 or 10 construct avoiding the flexible one. But with the cryo-EM, we don't need, we just purify and then we can see under the microscope. And also, but there is so many things. For example, there is so many issue that now developed in EM issue. For example, like noises in the micrograph. We call it like Einstein in the micrograph. So we can see the noises, but if actually it's not. But still, cryo-EM is still now people working on it, shifting from the X-ray. As I mentioned before, there is several function in cryo-EM. Cryo-EM itself is like called cryo because it is the protein itself to be in the cryo condition. So it's the EM in electron microscopy. So the atomic model building under below 3 Armstrong, it's we call it single particle analysis because there is another method for cryo-EM. For example, we have also cryotomography, and also the third one is micro ED. We have also like a crystal and we diffract, but not with X-ray, but with the electron with our facility. As I mentioned before, that the successful rate of a protein crystallization from the initial trial to obtaining the crystal is only typically like a 15% of what we have. This is the data. So that's why people working together to pass this bottleneck in the structural determination of protein using cryo-EM. So that's why also the pioneer of this got the Nobel Prize of working together with for cryo-EM. So what is cryo-EM, especially for a single particle analysis workflow? So we also we know that expression and purification already explained very well by Dr. Adrian, and we also have this is biochemistry optimization. For example, if you want to have some kind of the interaction with the ligand for drug or something else, and after that we put our sample in negative staining to make a contrast very good in under the microscopy, and we put our sample in the vitrification. It means our sample will put inside the grid. Later we will show you that how it works. And after that, this small very small grid we put in the EM electron microscopy that we now have Krios G4, the most sophisticated one in the world. So we can use that one for the structure determination, especially from BRIN and from Indonesia. And after that, of course, the data processing and model building that until now, Dr. Adrian still like teach me to work very well with the data processing and model building after result from data acquisition from our facility. So, but this is the truth that the sample preparation, vitrification, and screening optimization is the repetition work. For example, Dr. Putra also give us the sample last week, and then we need to do the repetition of the optimization from sample preparation, vitrification, and screening optimization. Because before we go to, for example, Krios G4, it is like consume like about 150 liter for 3 days. So we need a more efficient working with the Tundra before to do the screening optimization. So what we do actually like this. So in the vitrification robot, we call it vitrobot. It is like there is a paper inside this one to and then we put inside this 3 millimeter of grid, and this grid is very small, but inside this grid there is about like 200 or 300 mass that have the hole inside every 1.2 micrometer of the length. So later we can see. We put only, for example, in our laboratory, we design from 3.5 microliter until 4 microliter sample without crystallization. So after purification, you just bring us the protein, and we just put this one 3 microliter, for example, and the concentration also need no need very much as crystallization. We just need about 0.05 to 5 micromolar, but it is depend, of course, it's depend on the size. So the concentration, usually people working with the milligram per milliliter, please just recalculate based on divided by the molecular weight of the protein itself. So the protein will put here will be tapped by the paper here, vitrobot, and to take out the buffer, and the protein will be remain here. For example, this is the protein with many different orientation. So this orientation will be answer of how they do 3D determination under the electron beam. So this is the grid 3 millimeter. So if we can see, it is like a very huge, but actually very small, only 3 millimeter. But if we zoom in, they have a very consistent size of the mass. So this is, we call it grid square, but there is a hole inside here about 200 until 300 each. So inside this hole, there is very huge for the protein. If we zoom in, the protein inside is vitrified by the liquid ethane and under the many orientation of this. So one protein come out from top view, one another go to side view. It is the natural orientation. But after that, this orientation actually make a principle of the electron beam. It is the electron beam to the detector. So many picture of this, because this protein is only one type of protein. For example, we have we combine both protein complex A and B. We can put inside the tube and then put here. It's also same. We call it a single protein. And after that, with many orientation, we recalculate with AI. I think this is helped by Pak Rifki and BRIN, establish HPC, that very sophisticated one. Later we can show you how the AI help us in the structural determination, because the data itself is very huge. I have experience with this in collaboration with the Dr. Adrian. We have at one single protein, we got like a 5 to 7 terabyte. So it is a huge data only for one protein without any data processing. So it is like a crude data. We call it micrograph. So after that, we see here there is a vitrobot before. We just put the paper, and then this paper will be tapped the sample. The temperature should be very low, 4 degrees. Some protein like not very strong in the room temperature. So this is very small grid inside, and this is the condition. The humidity also should be very high, and we put the sample only 3 or 4 microliter, and after that they tap it very short time, and then we put it. Actually, it's like a very easy in seeing only like this, but in the in the lab is very, very difficult. So we need to optimize several times with this. And then after that, the protein trapped inside. So that what we do. So we just do the purification. After that, we do the negative staining. In BRIN, we have the operational standard using the Tundra with this, and also we put the vitrification and do the cryo-EM screening. And after everything's good, we go to the data collection with Krios 300 kV. So after that, the data processing will be like go to the HPC. Here there is people working with the HPC help me working with the structural determination, including Mbak Lina and also Mbak Maul working with this. We start doing the data refinement and also data validation. Until now, we have at least one that very, very good protein. It's about like 1.5 Armstrong. One of the best. So we now doing the validation and also the position. Of course, with help by our collaborator in NICE, Sensei Koga Sensei, also Adrian. So this is the very, very small. We put here the protein, and then we take only like a 10 or 12 grid square here, and then the AI, we call it APU. It is established by Thermofisher. Will collect the data. So it is very different with I work with Milan. It's they have a Glacios. It took like about like 4 days to take the data, but here in BRIN with Krios only like 4 days is equal for 4 days equal to be 4 hours in our facility. So but it is like a good investment. Still few people working with this with the protein purity. So this is we know I would like to say that the electron beam will beam the sample, and then the detector will see all the orientation of this. As Adrian mentioned before, there is a purification in AI. I call it like in Cryospark. We just like have the 2D classification. We select the classification, and this 2D classification will make the 3D of ab initio map that we can put inside our residue with molecular replacement and also passing with that and working with the 3D refinement until final high resolution structure. So this is the cryo-electron microscopy that work with the CPU. Maybe now Pak Rifki said to me that very good news that BRIN also invest like more working with the data refinement of the cryo-EM, because the cryo-EM data is one of the huge one compared to the sequence. I know that the sequence of amino acid from whole genome sequencing also NGS also very big, but please remember that the cryo-EM is data of the picture, so the 1,000 and million picture every protein, so it is like very huge, and with the metadata, so it is like consume a lot of the data processing. So this is the data processing I just put here. Later we can see under the microscope under real one. This is also real one, but I do the screenshot here. This is our cryo-EM facility, and then after micrograph we do motion correction and then do the 2D classification. For example, they do classification with iteration 40 times or also 20 times with the iteration moving inside, and then they classified its picture in the 2D classification, and then after that we select of it. For example, this one we will not select, and also not select, we just select several things, and then we have for example it's like only 2, for example, it is just an example, and then we can see do the 2D class again after taking this one as the reference. There is so many strategy with this, working with the AI. Balina working so hard with this, and then we like push the limit for the resolution, and then we have this the ab initio after homo refinement and do the sharpen of the electron density map from the cryo-EM. After that, we will got like this the data. We got it electron density map, the name. So as you can see here, it is not enough. Still do the refinement. It is many like sleepless night, I think, working with this. So the electron density map, we can see under the electron microscopy is only like this. No amino acid shown. But after that, we need to put our amino acid sequence inside here and do the refinement. For example, this one is going somewhere else, but the best one is like this, is going in the very, very good thing. So, but there's also like we call it PNIC and CCP for working with this. I'm happy to share, and also maybe like people in BRIN wanna learn of this. I'm really happy because I think until now no one worked with this. So I'm really happy to teach. So the section 4, I just want to share you maybe like only one my project that already published working with the cryo-EM. We call it spike body. So I think this project is not very interesting anymore because it is also already past the COVID-19 infection. But I want to show you it's about the cryo-EM thing. So the idea is we know that there is the spike that we need to block. For example, this one, many people block with the monoclonal antibody, but instead of antibody, we need to more stronger. So the idea is first we need to know the key residue of the conservative residue that conserve, not always like because it is RNA virus, so it's always moving to the mutation. So we do the selection of this with the study of many paper, and then we do the design the neutralization of we using nanobody. So we call it R and G because we just make it as like R as red, G and green because the idea is come like that, and then we make two of these. It's bind to only one because it is we know it is trimer. So this is we make it also trimer of this. So we to hit 3 times stronger. So the neutralization assay also show very good, except in SARS-CoV-1. So it is also can inhibit many of the variant of the COVID at the time. So this is the conclusion of what we have, but I will tell you of the story of this. We already published in Advanced Science 2024, I think. So we know that the entrance point, I think it is like not interesting anymore, I think. But I just want to show you that how the SARS-CoV-2 entrance in help with SC2 receptor. And the idea is we just want to block this interaction. So how we to block it? Of course, with mimic of this with the protein that are very similar with the affinity and also the character of the SC2 protein. So human antibody cocktail against SARS-CoV-2 at that time is so many because we just start and it is also now we identify the antibody that very strong, and then we also identify the potent neutralization antibody. So it's mean there is no heavy chain of this antibody, only the nanobody. We call it nanobody because the part of that the epitope of that bind. So we also identify the very specific one in the time. So we design this structure based on the bioinformatics at the time. What is the advantage of this platform? Actually, it's available from the start of new epidemic outbreak. So the idea is when the pandemic come up, we already know the epitope and just mimic the receptor, and then we put because it's very small, not like antibody is very huge and also have heavy chain. We just make the epitope that very active in the construct and then put inside our platform. So the second one is like I tell you that the lack of constant region of the antibody. And it is more efficient for neutralization because we can make so many antibody nanobody in one construct. So it is like will hit many time compared to the bigger, for example, antibody that that have a part not functional. So this is also we know that do not interact with AC2. This is also can implement to another like inhibition of interaction of another pandemic in the future, and also not specific for individual epitope. So this can be like a pan coronavirus specificity at that time. We work with one of the biggest hospital in Madrid. So we make a nanobody, and then the strategy is we want to make a trimer body to cover pan coronavirus, not only the SARS-CoV-2. So, but the idea is like the strategy at that time. How do we make it? But I think the idea is we understand this all by using the structural biology. So we take all the thing and then we analyze using bioinformatics. And also we see under the PyMol using this one, I make it using PyMol. And then the region of the spike, we also already identify that many of them is mutated, but many of them also still conserved. So do we design this the amino acid residue that very conserved, that can be like make the target of inhibitor for pan coronavirus. So after that we make a nanobody one, we call it, and then the most difficult one is we make the tree for, because it's a trimer. It is only bind to one monomer of the spike, so we make a tree of with the interaction of collagen. We call it have a trimerization domain. So, but until now, actually, NB2 is very difficult to make. But the thing is, the neutralization is very good at the time. And we will see how they bind. So the idea is we know the common infrastructure and protein engineering crystallography. Of course, also at that time, we start with the crystallography, not the cryo-EM, because at that time, very difficult to get the access in cryo-EM facility. Even like at that time, BRIN also don't have this cryo-EM. So we do the protein target identification. There is two protein identification here, like a SARS-CoV-2 spike protein, and the second one is nanobody that we make it. So we do the gene cloning and expression and purification and structure determination. The first one is the expression SARS-CoV-2 RBD, region binding domain, and we just buy the plasmid at the time, make a giga prep, and then put in the transfection and cell grow about like make it 2 million and express for 4 days. The expression very good, and we use the histolon using this histolon. As you can see, it's very small, but if we zoom in, it's very good and enough to make this a protein. So the size also correspond to this protein. This is the second peak, but the first peak, I think we delayed. So we took the fraction 32 until 39. After that, also we polish, because as Dr. Adrian mentioned before, it's very important. The protein that we need to determine is very pure. Some protein like make make its noise, for example, it's like make us trouble and very problematic when do another step. So we do the polish with using SuperDex size exclusion chromatography with high load, and we got only one peak. This is the second peak, is the salt peak that come out, and make sure that it's already come out all together in the sample. So we took the fraction number 32 and 35. As an information now, we in BRIN also we have like about 5 ACTA that can be utilized by BRIN and also people outside BRIN. Hopefully, it's like a maintained well in the future. So actually, in a short story, we established a protocol that also published for the high yield of pure unfolded protein with many strategy. The final concentration we got, it is the highest, only like final concentration, we got 12.5 milligram per milliliter, 50 microliter, only for 30 milliliter. So Dr. Adrian expressed maybe it is different because it's E. coli, it's 12 liter. So we using this, we can only 30 milliliter, but it is like hectonine 3 and SF9, it is different. So we do the stable analysis with thermal analysis. The thermal melting also very good. And the second one, we do the expression purification of the another protein, NB1, the complex of SARS-CoV-2 that bind. We do in the expression system with HEC293 because as we know that this nanobody, and we do the transfection with the PMAX system. We just permeabilize the membrane and then get in the plasmid, and then do harvesting until only 2 days, and then do the purification using strep tech. And we got this very small, but it's enough in the 38 clear Dalton. Of course, we also do the polishing with the high load SuperDex 35 PQ, and we got it. And then we compare, we inject. Actually, still it's already pure, but still there is. But we just polishing with the SuperDex. And in the final, we got like about 5.65 milligram per milliliter in 130 microliter. It is we do aliquot and then do the crystallization. But in the crystallization, we have no grow any at the time, unfortunately. So after many times, we don't have any crystal, and then we do the precipitation, and we got like still under optimization at the time. We plan to go to the diamond because at that time my boss is like one of the reviewer at in diamond in Oxford. And we do also the quality control assay using we see that using the spike hexa superrose, using also ELISA. And we do the cryo-EM facility. We have at that time this data micrograph. We we combine together about 4 hours in 4 degree, and then we put the vitrobot and this data collection. After that, we see easily it's like a helix, and we can see very easily. And at that time, I don't know how to do the data refinement with this, and we learned together at that time. I think Dr. Adrian still remember we wake up in the night to teach me with this yeah, from Japan and Madrid. And after that, we got from only 160,000 particle, we got 3.2 Armstrong resolution with electron density map. Isn't that really good at that time? Because nowadays in cryo-EM facility in BRIN, we got about like 1.5, 1.7. But in the beginning, we start to learn it's also very good. And we put the amino acid residue here and do the refinement. After do the art of doing the AI here with the cryospark with Reliant, and then we got a 3.2 Armstrong resolution. So this is how structure make us understand how they bind. It is actually bind very strong to the spike with this nanobody A and V, and every mono of spike protein, because spike protein have 3 mer, is bind every single thing. But the idea is we think how we can see the collagen of the interaction in every 3 of the trimerization domain. So we cannot see under this one because it's very flexible, as Adrian mentioned before. Taking the picture of the flexible one is very difficult if this one is collagen, because very soft to see. So we already published it in Advanced Science, maybe like it is one interest. But my role only, our lab only to determine the structure. The people working with this, with immunization and also the idea with the dendritic cell, they part. But our laboratory only to make understand how the structure, how they bind. So this is like the RBD one and the interaction of the nanobody protein. So this is our lab lead by Professor Ines J. Munoz, and also many people with the collaboration from USA and also from the Oxford in the Harwell and also Alba. So the last section is the introduction of cryo-EM. As many times I think I already told before, we have a sample, we do the vitrobot and do the negative staining with the screening with the Tundra. If the sample is good, everything, the concentration is good, the distribution also very good, we just go to take data collection here. So using this one actually can be like take many hours. And also with the Krios, we many like advantage using Krios. For example, the resolution, of course. The service is open to all the people in not only for Indonesian but also the region, because the Krios G4 is one of the sophisticated one. Until now, there is no G5 sold. We do the collaboration with Thermo Fisher, working with the G4 because our sample is very good at that time. They asked us to establish the G4 Krios, but until now, I think still making developing of the Krios generation number 5, sorry. So, but as I mentioned before, people working with this facility, of course, have the regulation that we need to apply, and also using ELSA also we can use. But we have the sample requirement. The sample requirement is first need to be like very polished thing. I mean the purity should be showing by the SDS page, and also maybe like only SuperDex one single peak. And the second one may be the size, and Mbak Ita also asked us about the 68 kilodelta in the tetramer. I think, yeah, Adrian also told us that it advised us to use the the crystallization. So that's why in the future we start to make the crystallization room in front of our cryo-EM facility that can be utilized for micro ED using cryos, because cryos also have the micro ED ability. So in the future, maybe the beta can have the expression protein and also purification. After that, do the initial crystallization for this. Just we can try. So this is the unpublished data we just deposited last week, yeah, with Adrian also. This is the more very like a sharp structure. So this is we can see easily the side amino acid tryptophan. As you can see, if you Google like a tryptophan have like this. So it is like we can see directly using the cryos itself. So if we zoom this one, we can see. So the resolution is now about 1.59. So this is very sharp. And the second one, this is also from BRIN. This is another protein. We cannot show you the name, but it's now the resolution also 1.69. Now doing the refinement by me and also Dr. Adrian working together with this. And also another unpublished data, protein Z, we call it, still very high, 2.71 Armstrong. But the idea is that is sharply from the upside, but the side one is very not good. So the orientation sometimes, the protein sometimes have a mono orientation. So only from the top, very good, but from the side they want to show us. So in the micrograph you can see we cannot see from the top, right? So it is only like from the side. So we cannot see the very good from the upside. So it is also the disadvantage using cryo-EM. And also we have the apoveritin standard, of course, is establishing this cryos. And also it is just showing you that we have also established for 2D data. For example, people working with this one is from our collaborator from US. Oh, sorry, yes, from US. And then this time they have a bacteriophage they give to us. And the later maybe we will do the single particle analysis of this and then do the 3D structure determination of this page. So also this is another one. It is from Indonesia, our collaborator that bind to this cell. And this also very interesting because if we can see this like only 50 nanometer, we can see clearly. So it is utilizing HPC led by Parifki in the beginning, working with to support cryo-EM research. But this slide is very made by Mbak Lina at the time. Parifki said like this nowadays they developing the newest storage and also data processing for cryo-EM. So the laboratory cryo-EM is located here in the first floor. We have this. The cryos may be here, and also the data storage itself from each is only 80 terabyte. You can imagine like only take one protein is already 5 terabyte. So it is like we're using fiber optic. They go to Indraja. So they establishing the Parifki this establishment, and then after that we analyze using communication with this, and then we using CryoSpark. I think it is like only the infrastructure that maybe like many of you people working with the computer know better than me. But the idea is now the bottleneck is protein production unit still just established. So after that we will establish the X-ray crystallography crystallization using the robotic that very sophisticated one, but I don't know, it's in the future. But now the protein production unit already established. Many people can use it and then hopefully can go to the cryo-EM facility. That's all from me. I think in the future, I think we can do the collaboration with us and then do the establishing of Indonesia Structural Biology Association. Thank you very much.
We are now open the second Q&A session. Whether you are a structural biologist or just curious about what goes into a cryo-EM pipeline, now is the perfect time to ask. We are waiting from the participant if Is there any question? Or maybe I should start again, Mr. Yudi. I would like to ask, this is like a really not heavy question. So basically, is there any chance one day the cryo EM price will be like the STS page and Western blot? You know, it's right now it's really pricey, right? So is there any possibility in the future that it will become like as we know before, the scanning electron microscope like SAM is such have the high price nowadays. It's literally we can access it easy in Indonesia. Is there any future for the cryo with the price?
Yeah, thank you, Pak Putra. So the idea that consume a lot for consumable for cryo EM is many. So I think it is like not only depend on the cryo EM field itself, but also related to the like a nano material. For example, making grid itself is very difficult because we need to make that one. The another thing is also the consumable for cryo EM is only like a grid, liquid nitrogen, and also liquid ethane. The other one is like making another thing is another part of field. For example, we need AI of this, of course, because AI is the one that can like help us to determine the structure from the picture. But also another consumable one is storage for the data. But I think in the future will be like a more cheaper because nowadays AI come up in good track. Maybe not as that much as now, because you can imagine like consume 150 liter of liquid nitrogen only for 3 days. So that's why usually I suggest to people user to combine together their sample in one run in 1 week instead of working like a 1 month operation of cryos. So this is like another thing. And another thing is like for me, I think in the future, tundra is the future, I think, because if we can see the aim of making tundra itself is not for negative staining, but for sure it is like to push the utilization of cryo-EM in the very cheap way, because the tundra only need 5 liter of liquid nitrogen, but it's only have 1 by 1 grid. So in cryos we have 12 grid itself. So in tundra only 1, and then the detector we have Falcon in cryos. In tundra we only have serta, but this limitation can like solve by, for example, if the protein is so big, so we can use the tundra it easily compared to the cryos, and maybe still cheaper than crystallization, I think, cryo.
Okay, we have now a question from Miss Ella. She asks about that kind of similar question to my previous question: how to obtain enzyme ligand structure using cryo, for example, membrane protein with the lipid. This is the hardest one. So, Mr. Yudi, can you give any insight?
Yeah, thank you, Dr. Ella. So question how to obtain enzyme ligand structure? Yes, it's the same. I just we already just solve the structure, the enzyme that have the ligand, but at that time we don't know. It is like a should be synthetic ligand, because I'm not a protein enzyme people. But Dr. Adrian told us to have the synthetic one, because if not, it's to be like consumed by the enzyme. It is like the easy way. So we put at that time we make, because we already established, we like a pride to say that our method is correct. We just put the ligand inside the buffer, and then also we combine together with the protein. We calculate the ratio of this using the molecular weight ratio. At that time, the easiest way is just to see the published paper, and then we mimic the concentration, and we incubate at that time about like 4 hours before putting inside the grid. And we got it, and then we saw the electron density map and its fit. Yeah, maybe Dr. Adrian will help me to answer because it's like an enzyme.
Yeah, I can add on. There are two ways, maybe like what Dr. Yudi mentioned, using synthetic substrate analog, which cannot be hydrolyzed by or processed by the enzyme, so you can capture the intermediate complex. Another way is you mutate the proteins if you cannot get the synthetic analog substrate. Then in that way, you mutate the important amino acid to alanine or inactive form, so they cannot process the ligand or substrate. Then in that case, then maybe you can capture the intermediate form by using the inactive mutant of proteins. Yep.
Yeah, thank you, Dr. Adrian. So I think it's like Dr. Ita also asked me, okay, when is precision facility ready to receive? Oh, still, we just start yesterday. So the idea is like we just start, and then we want to like establish, maybe like take several months to this, Dr. Ita. Or another idea is like collaborate with Adrian Sensei. He like to make this happen because Dr. Ita have the good protein working with. Yeah, maybe they like that.
Thank you.
Yeah, I think with that, we can conclude today's webinar. On behalf of the Aikman Molecular Biology Research Center BRIN, I would like to thank our esteemed speakers, Assistant Professor Dr. Chek Min Fei and Dr. Yudi Nugraha for sharing their research and perspective and time. We also extend our appreciation to all participants for your enthusiasm and engagement. As we continue to advance science in Indonesia, let us remember that great discoveries begin with the great structures, and great structure begin with the clarity. Thank you. Stay curious, and until we meet again, may your structure remain stable and your question always unfold. Good day and goodbye.
Thank you very much. Goodbye.
Okay, thank you, Dr. Putra. Thank you and all the speakers. Ladies and gentlemen, we have now reached the end of today's webinar. On behalf of the organizing committee, we would like to express our deepest appreciation to all participants for joining us today, and especially to our distinguished speaker for their valuable time, insight, and expertise. Before we officially close the event, we would like to symbolically present a certificate of appreciation to the speaker as a token of gratitude. Let us now display the certificate on screen as a symbol of appreciation. Mbak Rizka, maybe can you help me? Yes. To Assistant Prof. Chek Min Fei, or usually we call, I'll call by his close friend, Dr. Adrian. We present the certificate in recognition of your contribution as a speaker in today's webinar entitled Structural Biology for Health Research from Molecular Design to Structural Determination. Thank you once again for your inspiring presentation and meaningful discussion.
Thank you very much.
Okay, maybe take a picture. Okay. Next, to Dr. Yudi Nugraha, we present the certificate in recognition of your contribution as a speaker in today. Thank you once again for your inspiring presentation and meaningful discussion. Once again, thank you all for your attention and participation. We hope today's session has been insightful and beneficial. See you at our next event. Stay safe, stay healthy, and have a great day. Goodbye and thank you.
Thank you, Senpai.
Thank you very much.
Dr. Min Fei.
Thank you.
Thank you, Dr. Yudi. Thank you, Putra.